Welcome to another episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor.
Welcome, everybody, again to another episode of our show, bringing you another really fascinating guest today, helping to create a better tomorrow. Today, we have the honor of being joined by Dr. Arye Elfenbein, who is co-founder of a company called Wildtype biotech company focused on producing cultured seafood with a focus on cultivated Pacific salmon from fish cells sustainably, cost-effectively, that ultimately has all the nutritional benefits of regular salmon without the contaminants, without the mercury, the microplastics, antibiotics, pesticides, anything of that nature, or relying on commercial or fish farming. He was born in Israel, raised in Australia. Dr. Elfenbein combines his passion for medicine and unique connections in his childhood to the oceans to ultimately drive Wildtype strategy, their health and environmental mission. After he studied at Dartmouth and Kyoto University, where he got both a PhD and a medical degree, Dr. Elfenbein began his residency at Yale. He first trained in internal medicine, then completed training in cardiology. After his residency, he moved to San Francisco. He was working at the time with Professor Deepak Srivastava, from the Gladstone Institutes, doing things like cardiac transdifferentiation and morphogenesis and all sorts of exciting things on that front. The Gladstone at the time was also known for scientific luminaries like Shinya Yamanaka of IPS Nobel fame, and of course, Jennifer Doudna of CRISPR fame. And during that time at Gladstone Institute, his research focused specifically on cardiac regeneration following heart attacks. And a lot of that research has ultimately inspired him to apply a lot of what he learned in terms of the principle of stem cell biology beyond medicine to address another major problem, namely all the growing issues with our current food system and feeding the 8 billion people that this planet has. When he is not at Wildtype, Dr. Elfenbein continues to work as a cardiologist with a focus on critical care and lending his expertise and passion, of course, for patients in the ICU. We're lucky to have him with us today. Dr. Arye Elfenbein, thank you so much for taking the time out of your schedule to come talk to us for a while.
Thank you so much for having me.
It's great having you. It's a fascinating set of topics we're going to be getting into. I'd like to give you the floor for a couple minutes just to tell us a little bit more about your background story and a little bit sort of those early days of when you went in addressing sort of one of the largest healthcare problems we have in the sense of cardiovascular health towards focusing on an equally big problem, how we're going to feed the rest of the planet.
Yeah, I mean, I think like many of us in this field, it's not something that I would have imagined I would be doing when, for example, I was training in stem cell biology or training in clinical cardiology. But I think having worked in molecular biology at Kyoto University around the same time and in the same place that Dr. Shinya Yamanaka made the incredible discovery of how we can create iPS cells, essentially taking all of the moral issues around stem cell research off the table. It was an inspiring moment in many ways. I think there was so much potential to understand just how these fascinating cells that could grow into whatever they decided could proliferate faster than any other cells in the body, and there's so much to learn. I came back to the US and trained in internal medicine and in cardiology, and kept thinking about this question of how this could be applied even beyond the biomedical sciences. And it was around that time that I visited my childhood home of Australia, saw a lot of parts that were previously rainforest and now used for cattle farming, and thought about whether we could eat meat without eating animals, and essentially grow the same cuts of meat just outside the animal with everything that we learned from cells. And moved to San Francisco to work in Dr. Srivastava's lab. And actually it was what I was studying was a lot of the same principles of regeneration using RNA, the same RNA that is now the vaccine, but in the heart. And still this lingering question stayed with me of how we could use this, beyond biomedicine. Justin, who's been a close friend since 2011, he has a background in diplomacy and had worked in these very food insecure parts of the world like Pakistan, Afghanistan, and thinking through a lot of these questions of growing food insecurity. Together, we decided to give it a try. And San Francisco was a unique city at that time, in that there were places where you could rent lab benches by the hour. And that's what we did on nights and weekends. And that's kind of where this whole thing began.
Excellent. Excellent. And it's very interesting because I took a dive into PubMed, and sort of swam through some of your peer-reviewed publications from back in the day. And the really interesting thing here is, we normally talk about sort of cell-cultured foods, whatever they may be, and we think of sort of the public think, okay, here's some stem cells here, and here's whatever, the cow or the fish, whatever, and voila, there's some magic here. And the really interesting thing in your publications is that beyond stem cells, you were publishing on themes like branching morphogenesis. You published on angiogenesis. You published on aspects of the extracellular matrix. And I always wanted to bring this up because cells are not, whether it's a heart or whether it's a salmon we're trying to make, there's magic that happens in between the stem cells and that end product. Talk a little bit about how your work with some of the complex morphogenesis, the ability to form tissues and larger structures than the stem cell, sort of got you, shaped your strategy for what you're doing currently at Wildlife. And then we'll get into salmon in a little bit.
Yeah, I think the overarching theme for the research that I'd done in grad school and then afterwards was one of how cells communicate with each other. Looking at growth factors that they perceive in the environment or even create for other cells to know as that they should proliferate, for example, or start migrating. So when I was studying angiogenesis, or essentially the first stages of blood vessel development, there are two contexts where this is critically important. The first is in tumorigenesis. So as cancers can't really grow beyond a cubic millimeter in size unless they learn how to build their own blood vessels, essentially their own, roadways to bring in all the nutrients, and also to excrete waste. And a lot of people, and one of the pioneering scientists in this field was Judah Folkman, believed that we could curb the growth of cancers by essentially preventing them from growing their own blood vessels. And the other context where it was important was obviously in cardiac regeneration. So after a heart attack, a blockage in the heart, if we could have the heart regrow part of its own vasculature, theoretically, we wouldn't have as much of a need for bypass operations, and it would be just a more physiologic way to bring the nutrients to the growing to the recovering heart. And so studying this from the cellular perspective, from the very, very smallest units where this starts to happen, was a fascinating experience for me. The reason I went to Japan was for my PhD studies, was the lab there was focused on live cell imaging of these cells, and we could see which parts of the cells in real time were being activated, with certain molecules just being turned on, some being turned off, and it was just absolutely fascinating. Every time I would see how these cells interacted with their environment in real time, how they communicated with others, and not just with a light microscope, but also to understand the molecular complexity of these interactions. That is where, you know, it really all began for me, for wanting to better appreciate the complexity of these communication devices that they have. And so that, you know, when it came to the later work that you described in terms of the more of a cardiac specific function, that was very similar in that it was studying how cardiac cells can signal to each other to either become scar tissue after a heart attack or potentially regenerate as cardiac muscle tissue. And so that was the sort of overarching thing. What I never would have guessed was that so many of those principles have both so much similarity and are so wildly different when we think about them in the context of fish developmental biology.
Absolutely, absolutely. Well, let's get into fish. So, as I mentioned in the intro, the initial target of Wildtype is the cultivated Pacific salmon. I actually sitting here on the East Coast of the United States, and I don't know if so right now I have a sushi restaurant around the corner. I got a Great Deli down the street where awesome lox, and then a bunch of other restaurants that make macadamia encrusted salmon, all sorts of great. I love salmon, but I don't know if I'm eating Pacific or Atlantic. Talk a little bit about okay, so we have this 3 million or so metric tons of salmon that we consume each year. Talk about why the cultivated Pacific salmon, and tell us some fun facts about why this is the initial target of choice.
Yeah, I mean, you know, a few things. I'd say the fact that it's not clear to most of us what kind of salmon, where it comes from, and so forth, you know, just exactly what you described, is really, I think, where our troubles begin. So 80% of the salmon we consume in this country is imported. And I'd say something like 70 to 80% of the salmon that we consume also is farmed. And that's largely because our wild stocks of salmon have plummeted. It's been predicted that in the case of Atlantic salmon, that by the year 2050, with the current trajectory, that will become an extinct species, which is just, I mean, it is stunning to think about salmon, something we see everywhere, is so ubiquitous because of fish farming, could be extinct in the wild. And one of the issues with farming, which was really meant to take the pressure off the oceans when it was first, when it sort of reached its height of popularity, I'd say in the 70s and 80s. What we found is that there really were a lot of problems around sustainability. So there's a macro issue of sustainability, which is that if you believe in farming as a way to take the pressure off the oceans, still even until recently, most of the fish feed is dependent on ocean bycatch, and so the feed for these salmon were still coming from the ocean. The second issue is where we were seeing a lot of problems with bioaccumulation of toxins as well, because these fish were being farmed in such concentrated environments. There were a lot of issues of infestations, and so antibiotics became a common practice. And what we would often get was these environmental dead zones in these coastal waters where the fish were farmed. And so as our waters are unfortunately warming as well, there are fewer places where we can farm fish right now. And the future of fish farming looks like actually some of these on-land facilities. And, here I'll take a step back, because salmon are such a fascinating animal. I mean, they spend their early days in these freshwater rivers and streams, go out to the ocean for years, sometimes going thousands of kilometers and somehow knowing where to come back to exactly the same stream in which they were born and spending their last days there. To recreate something of such complexity on land, you need a freshwater environment. You need a saltwater environment as well. These waters need to be cooled, usually to something like 8 to 12 degrees Celsius. And actually, the largest one in the world is outside Miami. And so, thinking about the energy intensivity of something like that, it's yet to be proven exactly, how sustainable that is. And so, with all of the growing demand and the context for all of the issues we have in terms of keeping up with that, this was why Wildtype was founded. And we really do envision a future where wild caught salmon is an incredible product that people should be able to really command a premium from. People who are stewards of the ocean and care about the numbers of fish that they take out of the water. There should also be space for farmed salmon and for products like a plant-based salmon. And in our case, we wanted to create something that really had the same nutritional composition, but not just in terms of nutrients, but that these were actually salmon proteins, salmon fats produced by salmon cells, and that was the impetus behind pursuing the technologies that we use to grow salmon. And in terms of why do we start with salmon, in the U.S. it's the most consumed fin fish. Tuna is second. The second is that it's one of the most versatile fish. So when we think about culinary traditions, I mean, salmon is something that just like you said, we can have as lox on a bagel. We can have minced salmon in a salmon roll. We can have a salmon fillet, and that's something we don't see for a lot of fish, like I don't know, mackerel or halibut or something like that. And so that versatility, I think, really gave us an opportunity to showcase a lot of what cellular agriculture can do. And so this was, these are a few of the reasons why we began with salmon. I will say just that the last one is that when we thought about what we would create first, we didn't want to create ground beef.
Sure.
We felt like I don't know if the world needs more of that, and we wanted to create something with a lot of the benefits of, let's say, omega-3s, omega-6 fatty acids, and fewer of the saturated fats. And I think health was an important aspect. It was really an opportunity to create something that was free of all of these pervasive contaminants that are in our seafood system.
Absolutely. And speaking along those lines, I that's one of the things I was sort of thinking a bit about before our discussion. Obviously, salmon, aside from all those benefits you mentioned the nutritional, the omegas and so forth. It's a fatty fish, and once again, they got me thinking too. When we talk about the sort of the production, okay, we have some stem cells, we have interesting scaffolds. If you could talk a little bit about that, but challenges in terms of biasing production towards fatty properties. And once again, thinking to your cardiovascular expert, you don't want to ever make a fatty heart. And any interesting stories there in terms of lipidogenesis or whatever unique things go into that organoleptic and nutritional requirement of the tissue in this particular case?
Yeah, one of the things that maybe I'd just start with a microanatomy lesson that I learned was before we started Wildtype, I thought that the white striations that you see in salmon are similar to the to what marbling is for beef.
That's what I thought.
Yeah, and they're not. They're actually, for terrestrial animals like us, muscles are largely attached to bones, and that enables us to ambulate. And for fish, muscles are largely attached to other muscles, and so that enables sort of a lot of the complex maneuvers that their fins and tails will use. And so these areas of white bands or striations that are called myokama or myokomata. They're these little fibrous regions actually where muscles attach to other muscles. And so this is, it's an important part of the texture. It's also a very important part of the appearance. When you see something, when you see raw salmon, it's very immediately recognizable.
Yep.
And the first step was thinking through how to recreate that. It's also interestingly a place that that salmon will store some of their fats. So in ways that are maybe different from the how mammals do. And some of the fats are also stored outside of the cells, which does not happen as much in non-fish species. And so these are all kind of these fascinating learnings that that guided our work on both muscle creation and as well as adipogenesis, or the formation of fat cells and fat tissue. This is something that still hasn't really been done at large scale. In the biomedical industry, there hasn't been as much of a need to grow enormous amounts of fat tissue, for example. And so it's something that we're still very much learning on the way, and there are some very unique challenges in when we think about growing fat cells versus cells of other lineages within fish.
So Wildtype, as I mentioned in the intro, is definitely a company with a lot of firsts. We're just talking about sort of, you can be the first in terms of the cultivated salmon. Another first is just your scale up. You recently sort of christened this pilot scale plant, and once we have this figure that I mentioned at the beginning. So we have this 200 million tons or so of fish around the world, salmon, about 3 million metric tons of that. And the interesting thing, as I've gotten into this topic, looking back, say 10 or 15 years, everyone was talking about bioprocessing capability, and it was all going to be focused on, the monoclonal antibodies and the gene therapies and so forth. But it's really amazing how many organizations now on sort of the pharma side are developing these stem cell scale up capabilities to help take sort of the bench work through the process to get companies ready for these larger quantities, which are definitely going to be required. You've put together this plant recently, and you've moved a lot quicker than a lot of other companies in this sense. Talk about the new facility and sort of where that's taking you beyond the bench now, and sort of some of the scale that you're looking into practice there.
Yeah, I mean, the first thing I'd say is we certainly didn't know what we didn't know when we started this, and scaling up felt, like it would be a larger version of what we had gotten to work, on the bench scale. And to some extent that was true, but there are a lot of things we've learned along the way, because nobody has grown fish cells at the kind of scale we would need to have any kind of meaningful impact. And so, one of the first hypotheses we wanted to test was, could we adopt a strategy that was more like beer brewing than the pharmaceutical model of single-use systems, for example. And by that I mean, a lot of the medications, the drugs that are produced in the pharmaceutical industry rely on either fermentative processes or cell culture that are done in very expensive single-use plastic bags, sometimes $10,000 for one of these plastic bags, and then it's discarded at the end. And so this is antithetical to our mission for a few reasons. Right. First of all, it's use of plastics. The second is when you think about sustainability, and the final thing isn't so much our mission, but this is there's an intrinsic cost model there that is not tenable for our industry, as we're really creating a commodity product. And so we derived a lot of inspiration from the brewing industries and designed some of our own tanks in ways that were much closer to a brewing system. And so when people come to visit the pilot plant that you're describing in San Francisco, it feels actually very familiar in that sense. They sort of see these steel tanks that look like ones we see in breweries, and that's where the process begins. The thing is that that's actually just the first step. And growing a lot of cells in a tank is one thing, but these cells don't know how to spontaneously organize into muscle and fat and structures, and that a whole next step of how to get these cells to become a product is one that also hasn't been done at at any sort of meaningful scale in the past. And that's involved a lot of design as well, and sort of translating a lot of the findings we've had from bench scale, really what was a handmade process of combining these cells and a scaffold, and trying to envision how that could be done in a semi-automated and hopefully eventually a completely automated system at large scale. And it's something that's a journey we're still very much still on.
Exciting. So, Arye, so first cultivated salmon, first seafood pilot plant, another major first, the first cultivated seafood brand to raise over $100 million in venture capital financing. You have the Temasek organization from Singapore. You got the world's largest agribusiness in Cargill. You got some private investors, Jeff Bezos, Leonardo DiCaprio, and Robert Downey Jr. I never heard of them before, but I mean, where did you talk about? I mean, I've gone on venture capital road trips in the past and raised my share of funding, but talk a little bit about obviously this is cell culture food is very hot. I see a lot of brands, but $100 million is extremely impressive. Talk a little bit about your time on the road in bringing together this fascinating syndicate that you have here.
Yeah, I think the most interesting time of our fundraising journey was in the very beginning, and seeing what people's reactions were. And this was in 2017. And even at that time, it wasn't that long ago, but even at that time, people saw the promise of something like this, really believed that this is something that could be great, but we're very fearful of actually investing. And the trips that we'd make up and down the valley to various VCs and other investors, the common theme was like, wow, this is an incredible idea. This is something that could change the world. Let us know when other people are interested. And it was very disheartening in those early days. And we were very lucky to, through all of that, find incredible partners in Spark Capital. They were a team who just not only really believed in this and believed in our ability to actually make this work, but also were willing to take that risk, which truly is what venture capital is and should be. And then for our second round, we found just similarly just incredible partners in CRV. But until those two firms, until we had met those two firms and really like it was a very difficult and we process that I think left us both, Justin and me, pretty crestfallen after a lot of those meetings. Sure. And so, now where alternative proteins are certainly more part of the public discourse and people think of it as more of a hot field, it's I think a very different context to envision this. But it really was it was tough. It was very tough. It still very much is. I think that whether a lot of the underlying assumptions around being able to reach price parity, to dip below price parity, to be able to scale up, to have a meaningful impact in the world, have still yet to be proven. And we often think about electric vehicles in that sense. And the first Prius in 1996 was it was okay. I mean, it was nothing great, and it really took 25 years, and even further to think about the commitment in California, for example, to not sell any combustion engines from the year 2035 onwards. That's an enormous journey. And I think it's, the parallels are very similar in terms of what we need to achieve in cultivated meat and seafood.
And talking about risk for a moment, in addition, obviously, to dealing with the sort of the private equity and the venture capital community, you've also been very successful in sort of lining up distributors ahead of time. You have some agreements with a group called Snow Fox, which is involved in grocery store sushi bars, something called Poke Works, which is the casual poke bowl thing. How's that been? I mean, are these, and obviously nothing confidential here, but are these groups looking to, ultimately, once you have acquired the ability and the scale, are they looking to sort of eliminate all salmon and totally go with the cultivated? What's some of the stuff you're talking about? I know these are pre-distribution deals, but still, what are some of the things you can tell us about there?
But it's a great question, because I think it really hints at where people's minds are at, and I think that when we think about the conventional food today and the industries driving it, I think they see the writing on the wall in terms of sustainability, in terms of, for seafood, issues of human rights and slavery that still plague conventional seafood production. And it's been amazing to see people we wouldn't think of as interested in something like this see optimism in it and really believe in this and want to take that risk to mitigate a lot of these deleterious consequences that we all know is just is not going to work in the end in terms of overall sustainability. And so, in the case of Cargill, it took us a lot of conversations to really understand from their side how they see cultivated seafood emerging with respect to some of their own very fishing operations and fish farming operations, and so these are very complex but super interesting ways to think about how market forces can shape these over time, and eventually, like, it all sort of led to one place, and that was a recognition that our current system of producing seafood just simply wouldn't be enough. It just simply would not be able to catch up to the demand. And what you have then in the future, if you play that tape forward, is a world where only the super rich can afford great meat and seafood. Or we destroy the planet in the process of just, harvesting the last possible fish that we can. And I think that each of the partners that we've, met along the way has just seen, understands what that future, those two options look like, and has felt the optimism towards a third option where we actually are able to take the pressure off the oceans, are able to produce something that is free of all of these toxins, and produce them in a way that is more like beer brewing than dragging city-sized nets along the bottom of the ocean to try to just catch every fish that we can.
Absolutely. I mean, one other thing I noticed in your materials, and I haven't seen this, I guess, because you're much more advanced than some of the other cell culture food companies I've talked to, but you have written about the fact that, at the end of the day, once you have production and distribution, you still have the FDA there that, this is part of Food and, and Drug Administration. I've been in my career to the FDA primarily on the food side and the sort of the food ingredient side. What, if you could say, what are some of the things that you have to think about? Because obviously this is a novel food, not ingredient, not a drug, but nonetheless something that goes through their gauntlet of review. What types of things are you focusing on? What do you think you need to prepare for in terms of the FDA at the end of the day?
Yeah. We've been talking to FDA for almost 3 years now, and each time we have a discussion with the agency, I have to say I'm always really impressed by the thoughtfulness of their approach, and in many ways it really has been a process where they have asked to learn from us as we have asked to learn from them. And I can give an example in the very beginning. The first one of the first meetings we had with FDA, we were describing what our imagined approach would be, and it would be similar to the same standards of food safety as for conventional fish, and looking for some of the same toxins that might be present in fish. And we're describing some of these toxins, and the people on the call at the FDA said, wait a second, are you concerned that mercury is going to be in your product? And I said, of course not. What do you mean? And it's like, well, why are we discussing that? That's not one of the inputs, and it's not one of the and it's not in the end product, and it's not in the process. So why is this why is this even part of the conversation? And it completely changed the way that I, thought about what regulation for such a novel food looks like. And when we think about it in, within the current rubrics of how food regulation proceeds, it first of all makes more sense, and second of all, it allows us to really come to clarity much faster in terms of identifying all of those inputs, being very transparent about what goes into this, exactly how it's made, and all of the testing that's done afterwards, making sure that it basically will just be at the highest standards of food safety. And in that sense, what has emerged is a realization that what we create is actually not that controversial. Mm-hmm. So the main ingredient, salmon cells, has been around in salmon since, the beginning of salmon. Yep. And so that is not so much a controversial ingredient at all. Everything we feed the salmon cells are things that are present in our food system, like sugars and fats and, amino acids and so forth. And everything that is in our scaffold, these are all plant-based ingredients that similarly, have been around in our food system. And so it is the process that's new, and it's a novel food in that, it's a different way to create this. But mm-hmm. when we've thought through this and even, tried to envision what, potential consequences might exist from combining these things in this new way, and, discussing those and, writing them out and working with food safety experts, what we found is much more simplicity than complexity. And so that's maybe a long way to say that this process has been actually quite illuminating and a very very interesting one. And I think will hopefully also instill a lot of confidence for our future customers in terms of the thoughtfulness of the approach from both sides as we've pursued regulatory approval.
Excellent. Really excellent. Arye, what obviously you're very focused on salmon, but what's yeah. In the pipeline that you can talk about? And then I jokingly asked this question: if some billionaire came along and wanted you to make X, well, you have those billionaires already. But has anyone come along and said no? Let's make Y and Z, viper fish, giant squid, whatever it may be, anything odd that
Yeah,
anyone's to make.
we have gotten requests for dinosaurs, and I think that that's still much better served in great books and movies like Jurassic Park. But in terms of what's next, I think that there's so much to be done for not just ocean conservation, but for creating more healthy seafoods. Our focus really is on marine species, and so there are a bunch of others that we're already working on. And when we think through, what products we want to create, I think these are ones that, first of all, they need to be great food products. And if they're not, we're not going to have our impact, and we're not going to sway future customers in terms of choosing, a product like ours over conventional seafood. And it's one of the reasons why our focus has really been a culinary one from the beginning. So everything from the photos that we show of the products that we're now very proud of, to our website, like these are food focused. They are, not like lab experiments. These
Right.
are not science. Like, we are a food company at our core. And so these need to be products that delight customers, that people are really excited to eat. If people aren't choosing this over conventional seafood and it's just a novelty, that novelty will pass. And we won't be able to do anything for our oceans at that point. And so that's really the first criterion. The second, I'd say, is that these have to have some either ecologic or health impact. And so if we think about something like a threatened species like bluefin tuna, or one that is especially high in toxins like mercury, or is problematic for other reasons in terms of how it's harvested. For example, when you think about the harvesting of eel production, it's a fascinating industry in that we don't really understand how eels reproduce in the wild, and so it's led to a lot of very frank realizations of the complete unsustainability of the current practices. And so these are the guiding principles for the selection and product development. But in the end, they need to be foods that we're excited about, and that's what we're working towards.
Outstanding. It's a fascinating story. I look forward to, before the show, I went on the Snow Fox site, and I looked around for a location near me right now, but I don't think they're but whatever. I'm going to be looking forward to trying the product at some point.
Thank you.
Because you're obviously getting close to the finish line, and it's an extremely exciting story, and it's really it's elegant. It definitely fills such a major, think of talk unmet medical needs. It filled such a major unmet need on so many other levels. So it's really exciting, and I really wish you the best with all of this.
Thank you. I really appreciate your interest in this field, and and enthusiasm.
It is an exciting segment. I think you called it a niche a few years ago, but it's just so hot now, and I'm really excited to see all of this happening.
Thanks.
For everybody that is going to be listening to this particular episode of our show across the various podcast networks, or watching on our YouTube channel, again, you've been listening to Dr. Arye Elfenbein, co-founder Wildtype. Arye, I want to thank you for taking the time out of your schedule to come talk to us for a little while. Obviously, thank you for everything you're doing there. It's Wildtype, and as we like to say on our show, thanks for helping to create a better tomorrow through what you're doing. It's an amazing story, and once again, I wish you the best with all of it.
Thank you so much. Thank you for having me.
Good seeing you.